flow cytometry. So first in 1950, the first prototype of flow cytometer, which was not actually a flow cytometer, which was basically a cell counter, was introduced by Wallace Coulton. And finally, in 1967, uh, Heisenberg uh, first introduced the commercial flow cytometer to us. Now let's uh, describe about the Coulter counter first. So it was basically a machine where particles are being suspended in a conductive electrolyte solution and are drawn through a small aperture. And when a DC current is applied, the particles passes through the aperture and it displaced the same amount of saline equivalent to its size, which created a impedance which resulted in a voltage pulse proportional to the particular volume. By this uh, application, the number of cells of a suspension can be counted. Now, let's come to the modern flow cytometry and what are the principles of a flow cytometry and what is a flow cytometry basically. So, flow cytometry is nothing but a modern technology that measures and analyzes multiple physical characteristics like size, shape, internal complexity, and also fluorescence of a single particle, which are usually most of the cases are cells, and they they are being flown in a fluid suspension across an illuminated path. Now the most important thing of a flow cytometer is fluorescence. So now, first we have to understand what is fluorescence. So fluorescence is a property which is being uh, exhibited by some compounds or particles which are being called fluorochromes that absorbs energy from a particular light source of a particular wavelength and being activated and reaches to a excited state or activated state, which is a very unstable state. And due to the urge of regain of its stable state, in comes back down to its stable state and emits that energy which it have gained by the uh, light source in a form of light, which is a different wavelength from its absorbance. As an example, let's fluorescence, which is let's talk about FITC, which is being excited by blue light of wavelength of 488 nanometer, but it uh, exhibits green light, which of wavelength of 415 to 430 nanometer. 515 to 530 nanometer. Now, let's discuss about the basic components of flow cytometer. So there are mainly three. Uh, can you hear me, Rajveer? Hello.
Yes, yes, yes.
So there are basically three components of a flow cytometer: fluidics, optics, and electronics. These are the main three base of a flow cytometer. So fluidics are needed to focus the cells for integration and uh, in a suspension, fluid suspension, and optics are needed to generate and collect the light signals, and electronics are important to convert the optical signals to digital signals, and by which ultimately we can visualize and analyze the data in a computer system by a software. So these are three main parts of the components of a flow cytometer. Now let's understand what the fluidics of a flow cytometer works. How the fluidics of a flow cytometer works. First, the air is being pumped in the sheath fluid. There is a sheath fluid tank in a flow cytometer, which is uh, poured by a solution called sheath, and the air is being pumped in the sheath chamber by the air pump through a air filter, and due to which the pressure generated in the sheath tank, the sheath uh, goes into the machine or and ultimately to the flow cell, and uh, it flows through the flow cell. And now when we run a sample and operate it by the sample regulator, like running, pressing the run button, and uh, like selecting its flow rate like high or low or medium, the sample goes into the flow cell in the center of the flowing sheath fluid in the flow cell. And the sample the flow rate of the sample, the velocity of the sample is always high than the sheath which is flowing in the uh, flow cell, and ultimately it being heated by the cells being heated by the lasers, and the emitted lights are being detected by the photomultiplier tubes or the detectors, and ultimately the sample goes to the waste chamber as well as the fluid also, hey, sheath fluid also. Now you can say that there is a fluid which is already uh, getting like passing through the flow cell, and we are also uh, like running the sample which is also in a fluid suspension in the uh, flows, like and in the sheath fluid. So why it is not getting mixed? That is the one of the most interesting part of flow cytometer. Now we'll discuss about that. So there are two phenomena which are called hydrodynamic focusing and laminar flow. The Due to these two phenomena, the sample fluid doesn't get mixed with the sheath fluid in the flow cell. So what happens is, when the sample uh, fluid flow in a higher velocity into the center of another smoothly flowing stream, which is our sheath fluid, it gets focused along a central axis, because there are a difference in the velocity of the two fluids, the sheath fluid and the sample fluid. So due to which the sample fluid gets focused along a central axis of the sheath fluid, which is called hydrodynamic focusing or sheath flow. Now, when all the conditions are like exact and sample fluid flows in a central core, and uniform velocity is same, like uniform, like not it's getting increased or decreased, uniform velocity is flowing in a same velocity, then the two fluids doesn't gets mixed, and this is called laminar flow. Okay, now let's talk about the optics of a flow cytometer. So mainly there are two parts of the optics components of a flow cytometer. One is for excitation of the fluorochrome labeled cells or particles, and another is the uh, part where mainly collection of the signals are done. Now, in the excitation optics, there are mainly two uh, like subparts. First is laser, which is important to which is being used to activate the fluorochromes, and there are filters and mirrors which are being used to detect direct the emitted light to the appropriate emission block. And in the collection optics, there are fiber optic cables which uh, captures the signal, and it directs the emitted light through the filters, and the signals in the emission blocks goes to the appropriate photomultiplier tube. Now, uh, the lasers, there are more than two or more than three lasers in a flow cytometer, depends on the machine. So they are mainly uh, placed in a separated manner. Why? Because if they get merged, they can increase the background signal, and also they can increase the interference, and also they can result in spectral spillover. Like if like there are some fluorescence which can be excited by two lasers, not one lasers. They can be excited by both. Like suppose pi or p, which can be excited by a blue laser and also a yellow green laser. So if they are merged, the spectral spillover can be higher. Now this is a picture of an absorption spectra. So there are different types of fluorochromes are shown here. PRCP, FITC, PI, B, APC are there. Now let's take two random fluorochromes here, FITC and APC. Now FITC gets excited by 488 nanometer laser or blue laser, and it gives emission at 515 to 530 nanometer. And another fluorochrome, which is APC, that gets excited with the red laser at 632 nanometer and gives emission at around 660 nanometer. And which I was telling that in the before slide, that there are different fluorochromes also which can be excited through by a single laser, like FITC, P, PRCV, PI. They all can be excited by the blue laser. Now this is a emission spectra of different fluorochromes. Uh, you can see that FITC is there, PI is there, FT is there, APC is at 660 nanometer, and FITC is around 515 nanometer. Now let's know about the optical filters. Lasers, we have covered lasers. Now let's discuss about optical filters. So optical filters in a flow cytometer, which directs the emitted light to the photomultiplier tubes or the detectors, are mainly are of three types: long pass, short pass, and band pass. Long pass filters basically allows the light of a greater than a particular wavelength. Suppose it is written that LP 500, so it will allow only lights of wavelengths greater than 500. And short pass filters do the opposite, like they allows the light below of a particular wavelength range. Like Here 500 SG 500 is written, so they will only allow wavelengths of light below 500 nanometer. And the third one is band pass. So band pass is this type of filters mainly allows lights of a narrower wavelength, like it's written here 500 by 50. So they can uh, allow light from 50, that means plus minus 25, like 475 to 525. So they will only allow light as 500 by 50, its specification. It will allow only 475 to 550, 525 nanometer uh, wavelength. Now there are mainly two types of optical layout in a flow cytometer. This one is the old one, which is called herringbone pattern. So in this pattern, uh, let me first tell you that when the cells goes into the flow cell, and when the laser hits a cell, there are two types of scatters. One scatters, one part of the light gets direct transmitted through the cell and being captured in the opposite face of the laser, which is forward scatter or FSC signal. And the other part of the light, which is getting transmitted at 90-degree angle, these are called uh, like SS side scatter. And through from that signal, we can also detect can also now we basically detect the fluorochromes by different uh, filters and dichromic mirrors. So in this herringbone pattern, light gets transmitted through different filters. So transmission is the basic part of this herringbone pattern. Now, in modern flow cytometer, this type of layout is nowadays being used, which is called octagon or trigon layout. So this uh, layout basically is based on reflection. So here transmission doesn't occur, and here the longest wavelength is detected by the first PMT, and then the shorter wavelengths are being subsequent being respectively detected by subsequent detectors. And you can see here that in this layout, like octagon or trigon layout, the quality of signal is much more better than the transmission or herringbone pattern, because in transmission there are some loss of signal is there, but in reflection, there is not like not much loss of signal is there. So here in the octagon or trigon reflective layout, the quality of the signal is better than the uh, transmittance layout or herringbone pattern. Now, the third component of the flow cytometer, electronics. What is uh, the what are the parts, or what are the uh, things which are there in electronics of a flow cytometer? So mainly uh, the electronics, there are electronic parts in the flow cytometer also, and there are computer system also there, which is outside the machine, where from where we can regulate, or we can analyze, or we can acquire the sample. So the electronic system which is inside the machine converts the photons to photoelectrons, like from the emitting signal. Then they measures the amplitude, area, and width of the photoelectronic pulse. Now the uh, the photomultiplier tube was detectors amplifies the pulse either linearly or logarithmically and digitalize the amplified pulse. And finally, the digitalized form of the pulse can be visualized and analyzed in different formats of plots by a software and a computer system. So now, what are the different parameters of a voltage pulse? There are mainly three of them: height, area, and width. So, what is height? Height is being measured by the maximum digitized value, which is the peak of the uh, pulse, like from the base to the peak of the pulse. And what is area? Area is a sum of all heights, like from here to the uh, like end of the pulse, like start and it's going to the peak and is going to end. So from here to here, all the summation of the heights are the total area. And width, how width are being measured, which is measured by the ratio of area and height. So we, if we measure height and area, we can measure the width from our own. Now, how the optical signal or the analog signal is being digitized is shown here. First, the signal is like comes to the photomultiplier tube, and then it amplifies the signal in either linear form or logarithmic form, and then it digitize the signal and show us in a plot format. Now, what are the basic procedure of a flow cytometry experiment? So first comes the sample preparation and staining of the samples. So at first, the biological samples like blood samples, it may be, or it may cancer cell lines treated with any drug, or it may a transfected cell with fluorescence proteins. First, these kind of samples is being prepared. Like if it's a blood sample, it needs to be lysed. If it's a cancer cell line treated with a drug or transfected cell, it needs to be harvested and washed. Then this then like staining procedure comes, not for the transfected cells. Some place some in some experiment you can do also, like if you also want to check some surface markers of different wavelength, like it's tagged with different wavelength, so it can also be done. But staining is basically like for the blood samples or the cancer cell lines treated with any drug. So first you need to stain them. So, and which type of stain you are using, it depends on the experiment. Like if you are doing a cell cycle experiment or experiment to detect the dead cells, then you need to, or you are trying to uh, check the amount of the DNA after the, like which is being uh, destroyed after uh, treatment with any drugs. So in that case, DNA binding dyes like PI 7AD can be used. Or if you are uh, trying to see some markers like surface markers or any proteins inside the cells, you can uh, label them with an antibody which is labeled with a specific fluorochrome. So after staining, if washing is necessary, it depends on the experiment. Washing will be performed, and then the samples will be run in the fax machine. Then after running, or which is running is also is being called actually acquisition. So in acquisition, what happens? So when we run the sample in the machine, the cells move in a linear stream through a focused light source. Then the fluorescence molecule present in the sample gets excited by the laser beam of a specific wavelength and emits lights of a different wavelength which is also specific for that particular fluorochrome. And after that, the emitted light will be filtered and detected by the sensitive light detectors that are usually PMT voltage. And due to this, during this process, we may need to do some change in the settings, depending on our experiments, like changing the voltage of the PMT, gating of the particular cells of our interest, like population of our interest, and setting different type of plots, like which type of expression we're trying to see, like it's single expression or multiple expression. So these type of settings we need to do uh, in that time. Now after that, the analog fluorescent, which fluorescent signals which are being detected by the PMT or photometric tubes are being converted into digital signals. And finally, after getting enough data, like we done sometimes 20,000 events, sometimes we done 10,000 events, even sometimes we also run 1 lakh to 2 lakh events. So after getting enough data, we stop the run to analyze the sample. Now, in this slide, we'll discuss about the sample flow rate. I have previously said that there are three types of sample flow: high, medium, and low. So these pictures are actually the inside picture of a flow cell. So this is the actual flow cell you are seeing there here, and in the low sample rate, when the velocity of the sample uh, fluid is low, like 12 microliter per minute, the average width is very low. Average width is very low, not it's accurate, like 42.46 uh, yeah, micron. So now in low sample flow, it it is most preferable. Why? Because it maintains the hydrodynamic focusing, which is very necessary for uh, analysis, due to which only one cell can pass through the laser beam at a given moment, so we can get the proper data. But in high sample flow rate, due to wider width of the central core, you can see here the width is very low, very narrow, and here you can see that the width is very it's increased. And due to this, it allows more cell to enter the central core, like at the same time. So that's why, which results in error, which may results in error in signal at a given moment. So we can get high pulses or high pulse signal compared to the low sample flow rate in the high sample flow rate. So that is why low sample flow rate is desirable. Now, what is gating? So after acquisition, let's come to the analysis part. And first, it comes gating. So what is gating? Gating is a very essential tool in flow cytometry. So there, okay. So there are very there are different advantages of gating. So first is it sets the upper and lower limits on the type and amount of material that passes through. Like suppose we are only want to we only want to see the live cells. So we if we select the live cell and uh, uh, command the machine that we only check 20,000 even for the live cell, by getting that population, live cell population, it will uh, give the it will run the sample on the and acquire the 20,000 events on the basis of only live cells. And it the gating also provides a selection of specific population of cells for data analysis, like we what type of population, like live or dead or cells that are expressing a particular marker. So we can select that type of population of cells with gating, which help us for the data analysis. And it also allows the analysis of parameters specific to different subsets. Like if we are getting a population, we can all and it shows us that different markers, like if we are working with 4 to 5 markers. So there is one marker which is expressed in all of the cells. So if we get that cells basis on that marker which expresses all the cells, we can see what are the different type of markers, like other 4 markers, like we're using 5 markers, what are the other 4 markers and what are their percentage uh, in that population. And we can also like check the subsets, like another marker which is expressed in 30% of the cell, and like the another marker, like which is third one, that express 20% of the cells of that 30% of the selected cells. So we can also check the subsets of populations by getting. So you can see here, it is a picture of a normal blood, and forward scatter and side scatter is there, and only the lymphocytes are gated first, and they were uh, tagged with CD19 and CD3. And you can see that on after getting all of the lymphocytes, there are 17% of cells which doesn't express any CD19 or CD3, but 9% of that cells expresses CD19, and most of the cells expresses CD3, which is 74%. And suppose we are trying to see this CD19, there are some markers which are being expressed in CD19 expressing cells. We can also see that by getting the CD19 populations and checking other markers which are present in only CD19 expressing cells. Now, how many types of gates are there? These are different types of gates. So first, 3 types: rectangular, elliptical, polygon. There are 3 types of gates which are basically used for the same purpose. Like primarily get the population of any like experiment, but polygon is the most used among them. And then comes the quadrant gates. So quadrant gates is basically used for the multi-parametric experiments where more than one fluorochromes are being used. So by quadrant gates, we can differentiate between co-expressing cell and expressing single marker expressing cells. Like suppose we are doing experiment with two fluorochromes. One is FITC and another is APC. So there may be some of the population which doesn't express anything like in that in the previous slide, and with some of them may express only FITC, some of the cells may only APC, and some of the cells can also express both of the fluorochromes. So by these quadrant gates, we can separate different types of population according to their expression, like they are co-expressing or single marker expressing. Now the histogram regions, they are only applicable for histograms. They are very much being used in experiments where single uh, fluorochrome is being used, like cell cycle, Ross analysis, etc. Now, how a voltage pulse is being created in a flow cytometer machine? So, first, when a laser is when a cell enters the flow cell, when a cell enters the flow cell of the flow cytometer. One second, please. One second. Is it visible, Rajbir, now?
No, your slides is now not running. The slides are not running actually.
Uh,
Now it's visible.
okay, okay. Actually, my like internet just crashed, so I
Okay. No problem. Please continue.
okay. One second. Okay, so how a voltage pulse is being created in a flow cytometer. So first you can see when uh, this is a flow cell. When a cell or particle enters into the flow cell and just hits the laser beam, the pulse is being generated, and it's in the peak where when the cell is totally inside the laser beam, and it ends when just the cells leaves the laser beam. So that is how a voltage pulse is being created. So how many types of how many types of plots are there in flow cytometry? So first comes the dot plot. So what is dot plot? Dot plot is mainly mm, visualized by a single color, and there are many dots which forms up specific populations of cells, and there are color density plots uh, which is being uh, developed by the color is being developed by the concentration of the dots in that particular populations, like in dense population there is different color, and when the uh, population becomes lighter, the colors is different. And there is contour plot. So contour plot is basically when a, a 3D surface is being like expressed by a series of uh, constant z lines, which are called scatter in a 2D format. uh, they are called contour plot. So I am like going to discuss about what are the advantage of the contour plots. And there are another plots which are 3D plots. So 3D plots are basically plots which are being shown in three dimension or three axes are there, and they are being used to uh, show the relation between different populations or different expressions of the cells. Uh, Now, and another plot is also there, which is histogram plot, which are mainly used for cell cycle analysis or Ross analysis, which are mainly for single parameter experiments. Now, what are the advantages of a contour plot? So you can see that in the dot plot system, you can see that there are no different populations are there. It can be seen only a single population. But when you are getting the contour plot, there are two populations are there. Now, what is compensation? So there are different uh, fluorochromes. Compensation is also a very important part of flow cytometry. So what is compensation? Compensation is basically the spillover of signals due to the overlapping of emission spectra of different fluorochromes. So there are some fluorochromes which where their uh, emission spectra gets overlap, and due to which they give signal to the other fluorochromes channel. Like here you can see there are two fluorescent or FITC and P, and they're both excited by the blue light, but there are some regions here and here their signals get overlap, due to which you can see in this plot in the uncompensated data, you can see that the signal of the FITC is uh, falling on the channel of the P. That's why there is a false positive FITC and P co-expression population is there. But when the data is being compensated properly, that it reveals that the FITC, there is only FITC positive cells. There is no co-expressing populations are there. So that's why compensation is needed to properly analyze your data. And compensation controls are needed, like negative controls where uh, no tags are there, no antibody or no fluorescence are there, only unstained cells are there. And single uh, fluorochrome, like you are using three fluorochromes at a for a experiment, you need to stain the cells with single fluorochromes, like you are using FITC, PE, and APC, you need to uh, make different three tubes with only FITC staining, PE staining, and APC staining, due to which you can detect that if any signal of P falling on FITC channel, or any signal of FITC is falling on PE channel, and you can do the compensation by the flow software in the system. And automatic compensation can be also done. There are beats which are specific for different machines. So you can do compensation with the compensation bits also. Now, what are the applications of the flow cytometry? So mainly, uh, there are many applications are there, and which is immunophenotyping, to detect the surface markers and diagnose the disease of different like uh, different types of patients. Like if you are try to diagnose what type of leukemia or what type of yeah, cancer, blood cancer is there, is acute or it's chronic, or it is B cell specific or it's T cell specific, you can do with the immunophenotyping. And there are also dyes that bind to nucleic acids, like PI7AD, uh, which can also be used to different to do different type of experiments like cell cycle analysis, dead cell, life cell population analysis, etc. And you can also check the fluorescent expression of fluorescent proteins like GFP, YFP, CFP. And also you can use functional dyes like uh, annexin, uh, and also there are other also dyes which can be used for the different types of experiments. And you can also do cell count in single platform and double platform also, like using there are different dyes, calcine, which only calcinum is there, or which only stains the live cells, and cytoxerate is there, which only uh, stains the dead cells. And you can uh, add counting bits there. And you can also, there are also kits where there are counting bits already it's given there, so you can use these type of fluorescence or fluorochromes or kits to count as the specific how many cells are present in a specific type of populations. Okay, and you can also sort cells in by flow cytometer. So there are advanced version of a analyzing or only uh, analyzing flow cytometer machine. So in a sorter, you can get the cells that suppose you are uh, doing experiment with different type of reporter genes, like different type of fluorescence proteins, GFP, YFP, CFP. So suppose in your experiment there are three types of populations. One type of population expressing green fluorescence protein, one type of population expressing yellow fluorescence protein, and some of the cells expressing both. So if you suppose you need to collect the cells, they are expressing both of the fluorescence proteins. So by sorter, you can get the machine get the cells which are co-expressing GFP and YFP in your hand from the machine, which is a different machine from a analyzer. So this is just a representation of a cell cycle analysis which is being done by flow cytometry. So this is done by a program which is called PI, which only binds to DNA, and like DNA nucleic acid contents. It's also binds with RNA, so that's why we you staining with PI, you need to eliminate the uh, RNA with RNA's enzyme. And by the cell cycle analysis, you can detect if there, suppose there is some drug you're working with which can or cannot do the induce the cell cycle arrest at any of the checkpoint. So here you can see that there a drug is being used. So this is a control where all the peaks are normal. G0, G1 is there. This is your S phase and this is your G2M phase. So all are normal. But when you added drug which blocks at the G2M phase, so there is the, so you can see that there is a change in the G2M peak. So it's giving a higher peak than the G0, G1, which is opposite from the control. So this type of experiment you can do with the flow cytometry. So you can also analyze the apoptosis. So if any drug which can induce apoptosis in cancer cell line, you can detect that. And there are different procedures. Uh, this is uh, this is a procedure called annexin 5 and PI staining. So annexin 5 is a fluorochrome which only binds with the phosphoridyl uh, serine, uh, which which is to the surface of the, well, when a cells become apoptosis, the phosphorylation comes to the outer part of the membrane of the cell, and annexin bind in presence of calcium ion binds with that uh, lipid and gives fluorescence. So annexin 5 it's tagged with FITC, and uh, when your cell in the cell is in early apoptotic phase, there are no pore in the membrane. That is why PI cannot enter inside the cell and can't bind with the DNA and give the fluorescence. That is why in early apoptosis you can see that only FITC or only annexin 5 is there, and in the late apoptosis only both PI can enter in late apoptosis. There is pores in the cell membrane in late apoptosis phase, so PI can also enter there, and annexin 5 uh, and can also bind to the phosphorylserine. So both are expressed in the late apoptotic phase. And there is also a phase which is not shown here. Here some minute number of cells are there in the necrotic phase. So in necrosis, uh, the cells is already being accidentally dead, and there is no proper shape or proper presence of phosphorylserine is there, so annexin can't bind to the cell membrane and gives fluorescence. All, but there are many pores in the cell membrane, so PI can enter and bind to the DNA. That is why in the necrosis only PI is there, and in the early apoptosis only annexin is there, and late apoptosis both are expressed, or both are being captured by the cells. Okay, now let's come to the another type of experiment which is called immunophenotyping. In immunophenotyping you can detect abnormal cell population and categorize them according to their marker and detect the disease with the presence of the specific markers. So here you can see there are two different uh, uh, dot plots are there, which is CD45 versus SSC plot. So in this plot you can see this is a normal uh, uh, there is the plot of the normal sample. So there is no blasts are there. So mainly there are three populations: lymphocytes, monocytes, or granulocytes. So this is a normal picture. But when you come to the leukemia, here you can see that there is another population which emits dim signal than the uh, lymphocytes, which is there. So these are blasts which are expressed in which can be found in the leukemia patients. So from there you can get the blast cell population and categorize them by different types of markers. So these are this is a basic picture of a immunophenotyping of a AML patient, where you can see that MPO is positive. These are the specific myeloid markers. So in the blast, you can see here the blast region is gated, and they are shown in different plots where different markers are being used. Okay, so there are different markers which are myeloid specific, and we also used lymphoid specific markers also to detect if it's myeloid specific or lymphoid specific. So here you can see MPO, CD33, CD13, CD117, MPO is there, 117 and HLA-DR is there, 13, 33 is there. So these are all are myeloid markers present. That is why, and 34 is also is there, which is a blast marker. So due to which this patient was diagnosed with AML, acute myeloid leukemia. And you can see that the EA markers, lymphoid markers are negative, CD10, CD19, CD79A, cytosolic CD79A also negative there. So that is, it was that why it was a AML case. Now let's move on to the next slide. This is a picture of a BLL, where you can see that here MPO was positive, but you can see that MPO is not positive, and there are different other markers which is also negative, like CD33 is there with myeloid marker, which is negative. CD13 is also negative, and CD uh, 64 is also negative there, which is negative. Now here in this, it is a BLL case. That's why CD10 is positive, CD19 is positive, cyto CD79A positive. Like you can see here, cyto 79A. These are all lymphoid markers. CD79A positive, CD10A positive, uh, 19 is there. Here is 19 is there. 19 is positive. That is why it was diagnosed as a BLL case. Now this is a case of CLPD, chronic uh, leukemia. Here you can see that CD20, CD23 is positive, and anti-kappa is restricted here. So anti-kappa here, it is positive there, and CD200, 200 is positive, 43 is positive, and some other markers like TDT, CD5, CD10, these are also negative. That is why it diagnosed that as a CLL case, CLPD case. Sorry. Now, what are let's at last let me tell you what are the basic advantages. I will not elongate. So, what are the advantages? Advantages are there that flow cytometry is a very less time consuming process. Suppose you are doing when you are doing Western blot, it may take 1 or 2 days, but in flow cytometry it may take only 1 to 2 hours. So in there you are also checking the expression of some proteins, and you can also see here, so you can also check the expression of some proteins. So it is very less time consuming. And here in flow cytometry, every single cell is being analyzed. So the dot in the dot plot, you can see the small dots that there. So they are representing every cells you are running in the machine. So this method is very, that is why this technique is very accurate. And another thing is another advantage is this is a multi-parametric method. Like uh, if you are doing cell imaging with confocal microscopy, you can also uh, s- um, give the you can also check their multiple parameters like 2 to 3 to 5, maximum 5 to 6. But in now in this in a flow cytometers, you can easily check up to 16 to 17 colors. And another thing is in a cell image you can only see a specific phase. There are only 100 cells of there, but in flow cytometry you can uh, acquire or you can analyze uh, 10,000, 20,000, or even 1 lakh of cells. So that is why nowadays flow cytometry is being a very preferable technique to researchers and to the doctors also for diagnosis purpose. So that's all for today. I'm ending my discussion here, and thank you all again for your patience and presence here. Thank
Hmm.
you.
Thank, thank you, Dr. Ajit. It is very informative talk on flow cytometry. I wish you a very best and thank you very much for your time.
Thank you, Rajveer, for your good words. So, shall I disconnect now?
Yes, please.
Thank you. Thank you. Thank you all.